Welcome to this deep dive into isotope-labeled bioactive compounds, essential tools that enable us to trace molecular fates, elucidate mechanisms, and quantify biological transformations at atomic precision. Whether you're in drug discovery, metabolic research, or pharmacokinetics, isotope labeling offers unparalleled insight. Let's explore the science, the synthesis, and the real-world applications behind these molecular beacons. At their core, isotope-labeled bioactive compounds are chemically modified analogues of biologically relevant molecules in which one or more atoms have been replaced by their isotopic forms. These isotopes can be either stable, such as deuterium, carbon-13 and nitrogen-15, or radioactive, such as tritium, carbon-14 or iodine-125. The incorporation of these isotopes does not significantly alter the molecular structure or bioactivity of the compound, but does render it trackable by sensitive analytical instruments. In biological contexts, these compounds behave identically to their non-labeled counterparts, allowing for accurate monitoring of their pharmacokinetics, metabolism, target engagement, or degradation pathways. For example, isotope-labeled drugs are often employed to assess how a molecule is absorbed, distributed, metabolized, and excreted, collectively referred to as ADME profiling. Additionally, they are essential in elucidating enzyme-substrate interactions, receptor-binding kinetics, and intracellular trafficking. The precise tracking of isotopes provides a window into biological complexity with atomic-level resolution, and this makes them invaluable in both basic and translational research. There are two principal classes of isotopes used in bioactive labeling: stable and radioactive. Stable isotopes, such as carbon-13, deuterium, and nitrogen-15, are non-radioactive and thus pose no radiation hazard, making them particularly suitable for human pharmacokinetic studies and metabolic flux analysis. These isotopes are typically detected using techniques such as nuclear magnetic resonance, NMR, isotope ratio mass spectrometry, IRMs, and liquid chromatography coupled to tandem mass spectrometry, LC-MS/MS. Because they do not decay, stable isotopes are ideal for long-term metabolic studies, and as internal standards in quantitative bioanalytical methods. In contrast, radioactive isotopes such as tritium and carbon-14 emit detectable radiation, and are often used when extreme sensitivity is required. These isotopes can be monitored using scintillation counting, autoradiography, or positron emission tomography, PET, depending on the isotope type and its emission characteristics. For example, carbon-14 labeled compounds are frequently used in microdosing studies to understand early pharmacokinetics, while 18F or 11C labeled molecules are central to PET imaging in oncology and neuroscience. Each isotope comes with its own set of advantages and limitations, and the choice often depends on the experimental goal, safety constraints, and the required sensitivity and resolution. The synthesis of isotope-labeled bioactive compounds presents both opportunities and challenges, especially when dealing with complex molecular scaffolds. Broadly, synthetic strategies fall into several categories. One of the most common approaches is total synthesis, which involves constructing the entire molecule from simple, labeled building blocks. This strategy offers the highest level of control over the isotope position, and is frequently used when high isotopic purity or label specificity is required. Another important method is biosynthetic labeling, where microorganisms or plant cells are cultured in isotope-enriched media, incorporating the labeled atoms into secondary metabolites during natural biosynthesis. This is particularly useful for complex natural products or secondary metabolites. Exchange reactions are widely employed when labeling involves label positions, such as hydrogen atoms, that can be replaced with deuterium via acid-base catalysis or metal-mediated exchange. In recent years, late-stage labeling has gained traction as a practical and efficient route, particularly when labeling well-characterized drug molecules. This method allows for rapid access to isotopically enriched analogues without requiring complete resynthesis. For radioactive labeling, especially with short-lived PET isotopes like carbon-11 and fluorine-18, synthetic timelines must be tightly optimized to minimize decay losses. Chemists must also consider label stability, metabolic fate, and the preservation of biological activity when designing synthetic routes. Notably, the development of deuterated drugs, such as deutetrabenazine, has demonstrated that isotope incorporation can also influence pharmacokinetics, enhancing metabolic stability and extending half-life. The practical utility of isotope-labeled bioactive compounds spans across drug discovery, clinical pharmacology, metabolomics, and systems biology. In drug development, they are indispensable in ADME studies, providing quantitative insight into how a compound is processed by the body. This includes absorption rates, bioavailability, tissue distribution, and routes of elimination. Furthermore, isotope labels are critical in metabolite identification, where they allow chemists to trace specific atoms through enzymatic transformations. In mechanistic enzymology, isotope effects are employed to identify reaction intermediates, transition states, or rate-limiting steps. Isotope-labeled standards are also routinely used in LC-MS/MS quantification for bioanalytical assays, due to their identical retention time and ionization characteristics, which ensure high accuracy and reproducibility. Clinically, radio-labeled compounds form the basis for PET and SPECT imaging, enabling visualization of drug distribution, receptor occupancy, and disease biomarkers in real time. In nutritional sciences and systems biology, stable isotopes are applied in fluxomics and tracer studies to quantify metabolic pathway dynamics in vivo. These applications underscore how isotope labeling transcends traditional chemistry to become a foundational technique in biology and medicine. While stable isotope-labeled compounds are considered safe and generally exempt from regulatory classification as radioactive materials, working with radioisotopes requires rigorous adherence to safety guidelines, licensing, and environmental control. Facilities handling radioactive isotopes must be equipped with shielding, contamination monitoring systems, and secure disposal protocols for radioactive waste. Personnel must be trained in radiation safety and compliance with local nuclear regulatory authorities. From a regulatory standpoint, isotope-labeled drugs used in human studies must meet stringent criteria for chemical identity, isotopic purity, and radiochemical stability. In the United States, studies involving carbon-14 labeled microdoses must be filed under FDA's exploratory IND framework, which allows early assessment of human pharmacokinetics with minimal toxicological risk. Similar pathways exist in the EU under the EMA's microdosing guidelines. These frameworks are particularly valuable for evaluating candidate drugs with limited preclinical data. For radio tracers used in diagnostic imaging, good manufacturing practices apply, especially for PET tracers produced in hospital or academic radiopharmacies. Ensuring isotopic and chemical purity, sterility, and reproducibility is critical for patient safety and regulatory approval. In summary, isotope-labeled bioactive compounds are more than just chemical curiosities. They are precision tools that enable us to visualize, quantify, and understand molecular processes in biological systems. As synthetic and analytical capabilities continue to evolve, these tools will play an even greater role in shaping the future of biomedical science and therapeutic innovation. Thank you for joining us in this exploration of isotope-labeled bioactive compounds.